we're going to start our lesson this morning now, and I'm going to go through the whole plan of what we're going to be doing in this quite jam-packed session. So first of all, I'm going to start by talking to you about DNA, possibly the most amazing molecule on the planet.
Lucy Hutt is a science teacher at JFK School in Hemel Hempstead. In a specially extended lesson, Lucy will be taking her Year 11 top set through cancer and gene switching. They will also watch the film Cancer and the Genome and discuss ethical issues that arise. Finally, they will carry out a bacterial transformation experiment. Watching this packed lesson is Dean Madden of the National Centre for Biotechnology Education.
I'd like anybody to tell me what they know about genes. Dean?
Okay, yeah, they're made up of DNA.
Made up of DNA. Great. Do you know anything else about DNA, Dean?
It stands for deoxyribonucleic acid.
Impressive.
And it's got 4 bases.
It contains 4 bases.
But the thing which impressed me most was the breadth of knowledge they had. There were 1 or 2 comments that people made that showed that actually they understood a great deal.
Now that we understand how DNA codes for proteins, how the message is taken from the nucleus into the cytoplasm where proteins are made, I want you to think very carefully. What are all the jobs that proteins do within our cells?
Being scientists, we like to tell people about DNA replication and base pairs and protein synthesis and all of those things. And sometimes I think, well, maybe we shouldn't be dealing with those complex concepts. We should just go back to some of the simpler ones.
So in the light of Dean's comments, does Lucy feel that she has overloaded the pupils with information?
Actually, no. I don't think that I overdid it at all. I think it was quite a challenge to get from a very basic understanding of a gene and DNA to the extent that they understood that genes aren't switched on all of the time and that environmental cues are so important. Imagine you're a bacteria growing in a medium. Is it going to be sensible to have all of your proteins being made all of the time? What do you think the cell uses up when it's building a new protein?
Energy.
Energy, the precise answer. It takes energy to make new stuff, to make new proteins. So if you are a bacteria, it makes sense that you only have the enzymes that you need to break down a specific protein if that protein happens to be found in that environment.
Looking at the concepts that you might want to get across, I have a feeling that there are 3 of them. The first idea is that DNA is a recipe, a set of instructions. The second idea is that sometimes those instructions can have mistakes in them, mutations. And the third idea is that not all of the genes are active all of the time. Gene regulation is quite important. And it's that last message about switching genes on and off which frequently doesn't make it into the classroom.
Yeah, I think you're absolutely right. And it's down to the teacher to get that point across.
It is, yes.
And now I think it's time for us to talk about what goes wrong in cancer. Whenever we're talking about something such as cancer that can and possibly will affect in one way or another, possibly all of us in the room, we've got to be very careful that we speak sensitively about this issue. I felt it was important that the students did choose their words carefully because, you know, it was possible that there were people in the class with relatives, suffering with cancer or even having died from it, and they might be feeling particularly vulnerable, perhaps, to talk about it so openly. What do you know about cancer?
Genes are mutated.
Genes are mutated. Good girl, Emily. Anybody know anything else about cancer?
You can get lots of different versions of it, like skin cancer or lung cancer.
Right? It can affect every part of your body. Any other thing you know about cancer? Yes.
Does it occur when 2 bases aren't matched up with the correct partner?
Now you're getting clever, aren't you? We're now going to go on to view the film, which is actually going to be describing one particular form of cancer. And I should just tell you that the film is very graphic in its representation of the disease. You're going to see some rather ugly photographs of cancer on the skin, and I don't want you to be too upset by it, but rather because you will also be meeting in the film a woman called Sarah who's likely to die of her melanoma. I want you to be amazed at her courage. So we'll view the film, and then I'm going to welcome your contributions to our next discussion. I tried to prepare them for the film so that they would be able to see the positive aspects that the film also portrayed, both in that, yes, there's a risk with cancer, yes, there are things you can do to protect yourselves, and yes, there are people who get cancers that are going to kill them, and they themselves get the strength to cope with it. And that is something that the class should be impressed with. And I think they were. What was your main impression from watching that film? What will stay with you after the end of the lesson?
Probably the like the horrible pictures of the people with skin cancer, because that really was bad.
Was it worse than you had ever thought about?
Yes. Because most people can't see their cancer, but if you've got skin cancer, it's there staring you in the face.
Did anyone else have an impression that will stay with them from viewing that film?
The fact that despite modern science, they don't really know why enzymes make these mistakes seems that it makes it even more difficult to find treatment for it.
Absolutely. Ethics is important whether you're going to be a scientist or not, whether you really understand the science or you don't, because in the modern world in which we live, people are going to be faced with these dilemmas. Years ago, you had to go along and have a chest x-ray. And in those days, they were looking out to see if you had TB. Do you think this might be the future when you applied for a job, that they might ask you to have a genetic test to see which diseases you were potentially going to develop? What do you feel about that?
Well, it's slightly different, isn't it? Because the thing about TB is you can actually pass it on to other people. But say if you get melanoma, you can't obviously pass it on to anyone.
Do you think employers, or indeed insurance companies, should have the right to ask you about these matters? Yes.
I think that people should have the choice over whether they have a gene test, and that if they don't actually want to supply this information to insurance companies or to for their job, then that should be up to them.
I'd like you to understand that Sarah did not inherit this mutated form of the gene from her parents. It just arose within her cells, and it wasn't due to excessive UV. I want you to consider if you were a really, really brown person. Do you think it would be fair if the NHS said, well, you didn't listen to our advice. Now you've got cancer. You made that choice. Why should we waste money on you?
The NHS is still treating people who smoke, and they're still treating them if they contract lung cancer. So I don't think it's fair to discriminate on just one form of cancer. If you expose yourself to the risks, I think if they're going to do that, they need to do it across the whole spectrum of cancer. I think they need to consider really carefully about who exactly they refuse to treat if they're going to do that.
Does anybody have any other views about that?
Just because someone is tanned doesn't mean that they necessarily got melanoma from exposure to sunlight. It could have just been, like in the case from the video, naturally occurring.
And can I just say also that melanoma can affect whatever colour of skin a person has. You are still at risk of developing that condition. Just as the film showed you, the BRAF gene is mutated in 70 to 80% of cases. And in the case of the melanoma, the gene is forming a protein which is permanently switched on. And that's why you end up with cell division going out of control.
Having heard about BRAF, the class now understands that proteins can be in either a switched on or a switched off state. The same is true of genes. They can be switched on and off as required, and this gene switching is demonstrated by the practical. Before they start, the students must wash their hands with antibacterial soap to prevent contamination of the experiment with skin-borne microbes.
I want to keep this. I want to wear this into town one day.
We're not going to be doing experiments on cancer cells today, but we are going to be doing something rather amazing. What we're effectively going to do right here in the lab is switch on a gene in a bacteria.
The aim of the experiment is to demonstrate that a gene can be switched on by an external factor. Foreign genes are added to the host bacterium E. coli. One of the genes codes for green fluorescent protein, or GFP, and was originally isolated from jellyfish that glow in the dark. The GFP gene is under the control of a genetic switch that is triggered by a sugar. Bacteria grown without the sugar will not produce GFP and will not glow under UV. But bacteria with sugar present will switch on the GFP gene, causing the bacteria to glow. The experiment is readily available as a kit. It involves adding DNA to E. coli bacteria and then subjecting them to heat, which causes the bacteria to take up the foreign DNA. After several other steps, the bacteria are spread onto special agar plates, which will be incubated overnight to allow the organisms to grow. It's a complicated experiment, and timing of the various stages, particularly the heat shock step, is vital. But it's perfectly manageable if the groups are well organised. To make things run more smoothly, Lucy produces a step-by-step guide and assembles everything each group needs into one blue tray, a strategy which seems to work.
It ties in very well indeed, because there you've got bacteria which are genetically the same. You can have one petri dish with one set of bacteria, another petri dish with another set of bacteria, both exactly the same, but one of them glows green and the other one looks quite dull, completely different. And there, the difference is an environmental trigger. It's a sugar in the medium, arabinose, that switches on the gene. And there's a very graphic demonstration of gene regulation.
But what results are the students expecting?
This one will be resistant to antibiotic, but it won't glow in the dark because we haven't put arabinose, which is the sugar that's needed to turn the glowing on. This tray here, it should glow in the dark because it's got the sugar in it, and it should also be resistant to antibiotics.
Looking at the real results, Kieran's predictions are bang on. But the experiment they have started today won't be ready for at least 24 hours. So in the best traditions of Blue Peter,
It's time for us to see some results we did earlier this week. Okay, so these are the bacteria now that have taken up the plasmid, and they've also had the sugar present to switch on the gene for GFP.
What do you think about using practical investigations like this?
And I think they find it very motivating. I think the whole idea that they could be doing something that real scientists do in real laboratories, researching, medical issues or whatever, is a great motivator, definitely.